And we can start with our first speaker with its research. So the title of his presentation is Development of an Oral Immunization Strategy Against Filoviruses. So Juan García-Bernalt was graduated in biotechnology from the University of Salamanca and holds a master's degree in molecular biology from the University of Madrid. He earned his PhD at the University of Salamanca, and then where he was not working on vaccines yet. But he then transitioned for his postdoc, and his first postdoc was in the lab of Professor Michael Scott-Sire, that has helped us a lot actually to set up the next-gen group of the ISV, so we know him very well, at the Icahn School of Medicine at Mount Sinai. And now he reverted back to Europe since 2024. So as said before, he joined Professor Rafael Delgado's lab at Hospital 12 de Octubre in Madrid with a Sarah Borrell fellowship. So please, Juan, the floor is yours. We're all excited to hear your story.
Okay, thank you so much. Let me share my screen. Okay. So hello everyone, and I want to start by saying thank you to the ISV for inviting me to present on this next gen webinar series, which is just such a cool initiative. I'm Juan García-Bernalt. It was said, I work in the Research Institute of the Hospital 12 de Octubre in the virology lab of Rafael Delgado, and today I'm going to present one of our latest research lines, which is the development of an oral immunization strategy against filoviruses. So just to give some context, I'm sure many of you know filoviruses are filamentous pleomorphic envelope virus with single-stranded RNA negative sense of around 90 kilobases. And there are two major genera of filoviruses. We have ortho-Ebola viruses and orthomarburviruses. And within these two genera, there are up to 7 species that have caused disease in humans. The known animal reservoirs are fruit bats, and the structure of the virus is composed by 7 structural proteins. Today we will focus on 3 of them, which will be the glycoprotein, which is heavily glycosylated and is involved both in cell entry as well as immune response, both as a main target for the antibodies as well as for immune evasion. And we have the matrix protein, which is also known as BP40, that is the scaffolding for the virion, and the nucleoprotein. And these viruses can induce severe disease in humans with case fatality rates that can go up to 90% in an outbreak scenario. And today there are only vaccines available against one of the species of ortho-Ebola virus, which is thyre Ebola virus. So as I said, there are a few filoviruses known, and 7 of them are known to infect human, although only 5 have caused major outbreaks that are the ones here highlighted in blue. So from Ebola virus genera, we have Ebola, but we also have Sudan and Bundibuyo virus as the main cause of outbreaks. And in the Marburg virus, we have Marburg and Raben, which both have caused outbreaks. And this is important because vaccines that are developed now are not cross-protective between these filoviruses. And there are also some filoviruses that have only been detected in animals, especially bats. And I want to highlight Joviu, which was discovered here in Spain. And some of them, as Joviu, have been shown to have the ability to infect human cells in vitro. And all of these human infected filoviruses are overlapping in geographical distribution. So the epidemiological context is very complex in the filovirus infections. So today we have 2 vaccines license for using humans against Zaire Ebola, one based on VSV and one based on adenovirus and MVA, and the vaccines against other human infected filoviruses are in different stages of clinical or preclinical development. And we have candidates for Marburg, for Sudan, for Bundibugyo. But there is limited research on multivalent and broadly protective candidates. And also most candidates are intramuscular, which might not be ideal for a predominantly mucosal infection like is filovirus infection. So in this context, what we tried to do was to test if the oral administration of Ebola virus-like particle, which we like to call filoviral-like particle, because they have the classic filovirus morphology, can confer comparable immunity and protection against Ebola challenge when administered orally instead of intramuscularly. For that, we develop a FLP candidate which has three of the virus proteins, the glycoprotein, the BP40, which allows for the scaffolding of the VLP, and the nucleoprotein, which is not needed to generate the filovirus-like particle, but provides cellular immunity. And this is just performed by co-transfection of the three plasmids, so it's a very simple generation process. And then I'm going to present three of the experiments that we have done. The first, in which we try to optimize the sequence of the glycoprotein by removing some parts and adding a few mutations. Then I'm going to discuss the difference between oral and intramuscular administration. And finally, I'll discuss a protection study that we did with real Ebola virus in collaboration with Bernard Nock Institute in Hamburg. So first, what we did was try to optimize the sequence of the glycoprotein to one, improve trimerization and stability. And for that, we introduced two mutations in positions 577 and 588 in the glycoprotein sequence that had been shown before in soluble glycoprotein that they help trimerization. And then we did a version removing the mucin-like domain of the glycoprotein, which is very heavily glycosylated and is targeted by antibodies highly, but these antibodies are normally not protective. So it serves as an immune evasion shield. So we constructed these three versions and we vaccinated mice three times, 2 weeks apart, with 30 micrograms of these proteins. And we can see that the filovirus structures are conserved in these filovirus-like particles, as we expected. And then what we saw is that the three candidates follow a similar trend in terms of Ebola-specific IgG, although seems like the wild-type sequence leads to a little bit of a higher response. And when we separated between antibody subclasses in the mice, we see that both IgG1 and IgG2C follow a similar trend when we test in ELISA against the full glycoprotein. However, when we remove the mucin domain and we test the antibodies that are targeted to other regions of the glycoproteins, what we see, interestingly, is that the vaccination with the full sequence leads to more IgG2C, which should be a little bit more protective in this context, which wasn't really expected for us because we thought that maybe by removing the mucin domain, we will target more that region, but it wasn't the case. So we continue with the full sequence for the next experiments. So in the follow-up, then we compared oral versus intramuscular administration of the vaccine because we know that filovirus-like particles are remarkably stable, both in terms of temperature and not as much in terms of pH, but still stable. So we compare again with the same vaccination regime, but here, of course, as oral immunization needs a much higher immunogen dose, we use 10 times the dosing for oral, and this is still with no other adjuvant or other component in the vaccine. And what we see again is that antibodies against Ebola glycoprotein go up with the different doses in a similar way between the oral administration here in blue and the intramuscular administration in pink. And a similar trend is followed when we tested the glycoprotein without the mucin domain, and we can also detect some level of antibodies against the nucleoprotein. We then perform neutralization assays with Ebola glycoprotein pseudotypes, and we see again an increase proportional to the vaccination doses for the neutralization. And in collaboration with the lab of César Muñoz Fontela in Hamburg, we perform T-cell assays with nucleoprotein tetramer, and we can also see T-cell response after the vaccination. And in a third experiment, we tested protection with after challenge with real Ebola virus after oral administration, either one dose or 3 doses. And this was done in IFNAR mice that have immunocompromised interferon response, because immunocompetent mice do not get sick from Ebola challenge. So we did the vaccination, we collected samples after each dose, and then we collected the tissue from the animals as they reach the humane endpoint of the experiments. So again, what we see is that there is an increase in neutralization that is dose dependent. So here is after the first dose, there is no difference between 1 and 3 doses, of course, and after the second dose, there's already a difference in neutralization. And we see that in terms of protection, all unvaccinated animals reach humane endpoint by day 8. Animals vaccinated with 1 dose mostly reach humane endpoint, but there are 2 out of the 6 animals that survive. And when we vaccinate with 3 doses, we get a partial protection, so we get 4 out of the 6 animals that survive, and some of them even do not really lose a lot of weight. So in terms of survival, we get 66% of survival with 3 doses and 33% with just 1 dose, which is good for a not fully immunocompetent animal model. We see also that the viral titers in blood are reduced when we give 3 doses when compared to no vaccine or just 1 dose. And the viral titers in the different tissues, what we see is on the x-axis is the endpoint day where the tissues were collected. And of course, if they reach day 21, these are the survivors of the experiment. So what we see in black is the unvaccinated mice that have similar viral titers in every tissue, spleen, liver, lung, and kidney, to the ones that do not survive with 1 dose. And then the survivors with 3 doses reach humane endpoint later at day 10, and they have reduced viral titers. Of course, this can be just kinetics of the infection, but we think it's a very promising result. And we measure neutralization with real virus after the challenge, which is normally you need a much higher dose of serum to achieve neutralization compared to pseudotypes. And what we see is that there is a significantly higher neutralization in those animals that receive 3 doses compared to the ones that receive 1. And also we see within each group a clear difference between survivors and not survivors in neutralization. Finally, to compare all the measurements we did in these experiments, we perform a PCA where we can clearly cluster by vaccination regime, but we could also see clear difference between survivors here in the circles and non-survivors in the squares. Survival was mainly driven, of course, by reduced viral loads, but also by an increased neutralization. And the difference between 1 and 3 doses was also driven by a higher neutralizing activity in the 3-dose group. So in this context, what we thought is, okay, we may have a promising candidate for an oral vaccine, but this doesn't solve the issue of, okay, this is just a candidate for Ebola, so can we make a candidate that can broadly protect against other filovirus and that is easily administered orally? So we took inspiration on the papers that have been published recently for sarbecoviruses, where they constructed these mosaic nanoparticles where they express 8 different RBDs. And the rationale behind this is, okay, maybe we can select B cells with BCRs that can cross-link between non-identical antigens by binding very conserved regions among them, so that could not be selected otherwise with a homotypic nanoparticle. But of course, we need to apply this to our filovirus-like particles, and it's quite interesting for filoviruses because all the information we have from monoclonal antibodies that have been derived either from patients or from mice that are infected or vaccinated with Ebola or with Bundibugyo show that here in orange there is not really cross-binding between ortho-Ebola virus and Marburg virus, or just very little cross-binding. So maybe by introducing this mosaic composition of the nanoparticle, we can get there. So we design new glycoproteins with a modified transmembrane and domain and cytoplasmatic tail, where we include the transmembrane domain from Ebola glycoprotein into other filoviruses, such as Bundibugyo, Sudan, and Marburg glycoproteins. That way, in an scaffolding of Ebola, BP40 and Ebola nucleoprotein, we could attach the different glycoproteins from the different filoviruses, and even we can generate these mosaic filovirus-like particles. And as you can see with the ELISA data, with the original transmembrane domain, we didn't see any expression of the heterotypic glycoproteins in the surface of the filovirus-like particle, but when we modified the transmembrane domain, we could see all 4 glycoproteins simultaneously in the filovirus-like particle. So when I took this webinar, I was hoping to show you the results from this immunization experiment, but unfortunately we had to delay, so it's now ongoing. So we are going to compare these mosaic filovirus-like particles, both intramuscularly and orally, with the combination of the cocktail of all the filovirus-like particles, and see if we can generate broad antibody response and neutralizing response against the main human infecting filoviruses. And we are also adding an adjuvant to see if by broadening the response we get a lower overall response. So maybe we can even push it further with an adjuvant. So just to conclude, we did some modifications of the glycoprotein sequence to improved trimerization or removed the mucin domain, but this didn't improve the vaccine response. Oral administration leads to a similar humoral and cellular response to intramuscular administration, and oral vaccination protects partially in IFNAR mice in terms of morbidity, mortality, and viral titers. And that modification of the transmembrane domain and cytoplasmatic tail of the glycoproteins allows to generate mosaic filovirus-like particles as a potential panfilovirus vaccine candidate. I just want to finish by thanking all the people that has worked in these experiments, especially from our lab, Fatima Lasala, did the initial experiments, and Hugo Morgado Ruiz, which is a very talented PhD student that now is doing all the heavy lifting in the experiments. Also the people at César Muñoz Fontela's lab, especially Beatriz, who led the animal BSL-4 experiments, and our collaborators in the National Center for Biotechnology that help us with monoclonal antibodies and with protein production. And yeah, I just want to thank again the ISV for allowing me to present today, and I'm happy to take questions rather now or at the end of the webinar. Thank you so much.
Thank you so much, Juan. This was super interesting. So I wish you, first of all, the best of luck with your study, the last one. I
Thank you.
hope it will come out as expected or as we would like. So first of all, we can ask maybe one question to Juan before going to Inès. So please, if you have questions, just write them down in the Q&A below so that I can ask them to the speakers. I see that someone has the hand raised in the attendees, so please just write down in the Q&A session. If it's not asked now, it will be asked later at the end of the Q&A. So for the moment, I see one question. Ebony, you have a question? Here it is coming out. Did you have the chance to measure neutralizing antibodies in BAL or Langwash?
No, so we just did this as proof of concept experiments, but we are now getting mucosal samples from this new experiment to see if we can both detect IgA, which we didn't measure in these first experiments, and also see neutralizing activity in other tissues. Yeah.
Thank you very much. So, okay, I will just ask another question, and then we will move to the end to the Q&A. So there is a person that you know that asked a question. So Michael, in the Ebola challenge experiment, do you see strong disease modulation, cytokines pathology in the survivors?
So we didn't measure cytokines, or we took, there are some tissues stored, but I don't think we have made any histopathology analysis to date. But yeah, there's definitely something interesting to look into. Yeah.
Thank you. So, okay, let's switch to Inès. I see there are two other questions. I will ask them. Just wait until the end of the Q&A. So,
And like,
sorry, I forgot. Sorry.
So yeah, it could potentially be when it was done in mice, it was in the ears, because that's where it's kind of like easier or handier. And in the pigs now is in the leg. I guess it could potentially, that's part of like the mechanism of action. I guess we're still trying to elucidate, but it would be something nice to try.
Thank you. Then for Juan, I have also here a very nice presentation. Do you see a different immune response profile after oral vaccination in IFNAR mice compared to immunocompetent mice? In other words, is interferon important for your filovirus VLP oral vaccination?
So the honest answer is we don't know really. So we did the protection experiment only in IFNAR, but as they do not have interferon response, we did not measure T cells. So we don't really know. We see neutralizing responses that are comparable, and antibody responses are comparable between the C57 and the IFNAR. But in terms of other arms in the immune response, we just don't know.
Thank you. So another question for Inès. Wait a second. So the patch did not completely abrogate anti-vector responses. Have you considered evaluating anti-vector responses after multiple boosts, since traditional flu vaccines are repeatedly administered?
Yeah, definitely. That could be something to be looked at. We're a bit limited as well for like pigs are not like mice, not that easy to handle. So very long and extensive multi-dosing experiments, like studies, are difficult to plan. But definitely it would be something to look at, and it could also be looked in combination, like not just only using patch, but maybe combining using IM and patch, using the advantage that patch decreases, even though it does not completely abrogate the anti-vector response. We could like play from that.
Thank you. Then I have a question for Juan. So one of the things is that you said that your platform is not is a bit pH sensitive, right? So have you considered going for a formulation at one point, and how would you tackle this?
So we have thought about it. So at this point, we really do not know how the mechanism works for the oral induction of the immune response. So we will look into it, and then it's hard for oral vaccines for formulation, also for adjuvants. This is not as well characterized. So we are working on first see if with this PLP just by itself, because it seems to produce a very strong immune response. So maybe we don't need anything else. And if not, we are working and contacting people that are much more of an expert compared to us to do these formulations. Yeah.
Nice, thank you very much. So I have another question to Inès. So what is your explanation for an improved ratio of insert versus vector response in the patch vaccination?
Yeah, I think that's similar to the question before. I guess like why the anti-vector response is better in the patch, well, lower. Yeah, we don't really know the mechanism behind it. As I said, might be the way it is released into the body. Maybe the could be also a matter of like kinetics,
Yeah.
but still looking into it.
So since I went for a similar question of what we had before, so we have some people that are curious on how you load the vaccine into the DMN patches.
Oh yeah, well, that's it's all about the formulation as well, and then we basically need sugar and polymer for the structure of the needle and to protect the vaccine, and then we just load it into a mold that has the pores, and we load it into the pores. We let it dry, and then we remove it with a tape, and that's the kind of image I showed before. You could see like the tape with the small needles on it.
Thank you. So maybe I have a question for both of you. Can you provide other vaccines that could benefit of your platforms?
So I think
Other models.
yeah, for us it's a bit complicated because it's like the filovirus particle itself. So I think outside of filoviruses, I don't know if it makes sense. Also, probably the particle physical characteristics and the robustness is not the same for other viruses. But I think the concept of the mosaic vaccines is, of course, applicable to many highly variable viruses and other pathogens, and it has been used before. Yeah.
I think the patches, what we are aiming for, like really they could be applied for many vaccines. It's a matter of formulating them and also like really seeing what is this profile of immune responses that they are offering to see which ones would be more advantageous. But it really also comes to the accessibility advantages that the patch offers. So really in like any vaccine that could benefit, and it should be able to be implemented in the patch, because the patch is really like a carrier, like a delivery technology. You just need to put the vaccine in and like, yeah.
Yeah, sure. Thank you. So I think we will go with 3 more questions and then time will be up. So thank you very much for all the responses. You do both have a lot of questions. So, so how consistent, robust is the manufacturing process for the microneedle dermal patches in terms of microneedle integrity as well as consistency of dose between microneedles full patches?
Sorry, I think it got a bit cut for me there.
So, how consistent, robust is the manufacturing process for the microneedle dermal patches in terms of microneedle integrity as well as consistency of dose between microneedles full patches?
Yeah. So yeah, that was something we had to develop, especially to adapt the dose needed for the pigs. But then once we have the protocol, it's quite consistent. It's still a manual fabrication at the moment because we are doing a small preclinical trials. But we are seeing quite consistent infectivity results post fabrication from the patches for the adenovirus vaccine. And yeah, consistent doses.
Okay, thank you. So I think I can ask one more question to Inès, and then I thank you all also from the public to have asked so many questions. It's very nice to have this amount of exchanges. So a subset of HA binding antibodies have no neutralizing activity in vitro but are protective in vivo, which has been linked to FC-mediated functions. Have you tested in the microneedle elicited antibody have increased FC-mediated antibodies function?
That's what we are trying to look at. So that's what we are trying to establish. There's not as many established methods for pigs as there would be for other animal models or for humans. So that's why we are still working on setting them up, but we are really interested in FC functions because, yeah, I mean, we know that other than neutralization, they are quite key. So we're specifically interested in the antibody-dependent cell phagocytosis. So hopefully soon we'll have some answers regarding the FC functions of antibodies, but not so far.
Thank you very much. So, thank you very much for both of you. It has been a great webinar. We had a lot of attendees. We've had a lot of questions, so great presentations, a lot of success, and I wish you both a lot of success with your research. Thank you again for accepting to participate. If you're interested in joining our next gen ISV, you will be always welcome. And yeah, for everyone else, let's see each other again on the 29th of April for our next webinar. It's going to be a very interesting one as well. So thank you. And thank you also for listening and for participating. And have a great evening, morning, afternoon, depending where you are.
Thank you very much for the invitation and to everyone for listening.
Thank
Thank you,
you.
everybody.
Bye bye.